Let's discuss a little bit about fermentation. Nice diagram of a variety of glucose usage, right? So we've got all this squared away. Glycolysis going this way, gluconeogenesis going that direction. And gluconeogenesis, right? There's some glucogenic amino acids which can go into pyruvate. There's lactate which can go into pyruvate, right? So recall in another lecture we talked about pyruvate, is used to make glucose. But where did the pyruvate come from? Right? Lactate and amino acids, which are the common ones. What's the other one? Oxaloacetate, right? Oxaloacetate. So, but what we're talking about now is shunting off and making lactate. So under conditions of low oxygen, we can't run the citric acid cycle as much. Okay, so the citric acid cycle will create NADH, which then goes to the electron transport chain, which we'll see. So lactate is something we do, and yeast is something that yeast will make ethanol for fermentation. And this step right here is often called the 11th step of glycolysis because even when you are, have a lot of oxygen and you are running the citric acid cycle, there's still a little bit of this happening, right? So remember, there's never 100% and 0% of any of these. There's always some amount of reactions going on. Okay, so glucose to pyruvate through glycolysis. Pyruvate dehydrogenase complex using TPP plus other cofactors converts pyruvate to acetyl coenzyme A. Acetyl coenzyme A feeds into the citric acid cycle. There's a whole lot of molecules that go in and out, but if you just look at when the citric acid cycle is running for the purpose of energy, it is using NAD plus and kicking out NADH. NADH will go into the mitochondrion where it will then be oxidized, create a proton motive force, and use that proton gradient to form ATP through ATP synthase. So when we look at just that, if you're running all of your glucose to fully carbon dioxide, right, through glycolysis, the pyruvate dehydrogenase complex, and the citric acid cycle, what we see is that we get some of those ATP from substrate level phosphorylation. That's why that goes to just ATP. Same thing here, we get some GTP out of citric acid cycle. But under oxidative conditions, most of our ATP comes from the NADH and the FADH2 through oxidative phosphorylation in the mitochondria. However, when oxygen is low, you start running, oxygen is consumed very, very rapidly. Your system has not started putting in a lot of oxygen yet into your bloodstream, so there's a period of time where your muscles go low oxygen. And so we need to still generate ATP. In that case, we'll generate ATP from glycolysis only, mainly only, never 100%. Okay, so to do this, we need to regenerate NAD+. And so if you're going to get all of your ATP, right, so one glucose molecule, 2 ATP, if this is now where you're going to get most of your ATP in muscle, well, you need to regenerate that NAD+, because you'll start generating a lot of NADH, you build up NADH. If you're not running the citric acid cycle, you're not regenerating your NAD+ then, right? So that's why we undergo homolactic fermentation. So when people say, that you get low, anyway, I'll just leave it at that. So that is the function of that. And so what's my next slide? Yeah, so it's not all about us though, right? So look at yeast, right? So all of the ethanol that exists pretty much is through fermentation from yeast. So whether it's beer or wine or whiskey or vodka, right? If it's ethanol, it came from fermentation. And so this is the pathway for that. Why is the yeast using fermentation? Well, if you've ever made beer or wine, you know that you put it in like a carboy or a bottle, and then you put an air seal on it that's got water in it that allows gas out, and it burbles through the airlock, and it doesn't allow the air from outside in. So the yeast are anaerobic at that point, so they're not getting any more oxygen. So how are they going to live? They need ATP, well, through glycolysis and ethanolic fermentation. That's how, right? So no more oxidative phosphorylation for yeast when they're making wine or when they're making beer. And it doesn't just stop there. Look at the different types of molecules that bacteria can make. So as a matter of fact, this is propionate, but there's a bacteria on our skin that it's called Propionibacter, and it produces propionic acid, and that lowers the pH of our skin, which makes it inhospitable for some bacteria. So this right here, this is an example of many pathways of, a few pathways of many that exist of microorganisms that either do this only when there's low oxygen, or there's some bacteria that are facultative anaerobes. Like oxygen will kill them. They do not have that oxidative phosphorus. They do not have oxygen as a terminal electron acceptor. Now in that case, they can use other things, and it's not just fermentation, but still. So let's see. So benefits of fermentation: provides ATP very fast. It's not efficient, right? You're not oxidizing it completely, but you can get it fast. So that's why in our muscles, right, it works. The products have their own fates. Many microbes, so like when these, in soil, for example, when bacteria releases propionate, well, that is a carbon molecule that other bacteria have evolved to use as a carbon source. So there we go. Some obligated anaerobes rely on different fermentations, as I mentioned. And so not only do we use this for us, but we can also take pyruvate from other mechanisms, not glucose, right? Because if we need to make glucose, we can make it from lactate. So we'll see that lactate can be made from a variety of other things. So the liver uses lactate to make glucose. Okay, so important step. So let's look at lactate dehydrogenase. So we have this, right? We've seen this slide before. This is one of the ones we saw a couple units ago. And so what we see is that a hydride transfer to pyruvate, converting that into an alcohol. This was my example for us of, if you look at a carbonyl to an alcohol, what's your first guess? Your first guess should be NADH, because that hydride is going to transfer to there. Both of those electrons are going to go, it's going to kick up and form a hydroxyl group. Likewise, if you have a COH, right, a CHOH, and it needs to be formed into a carbonyl group, what's your best guess? The best guess is that you're going to use NAD+ to pull that hydride off and form a carbonyl group. So we haven't left that yet, still know that. And then this is showing a little bit different version of the mechanism here, right, where the hydride is being added, and then it is pulling off a proton from somewhere to form lactate and NAD+. Now, over here, alcohol dehydrogenase, yeast will then add the hydride to this carbonyl group of acetaldehyde to form ethanol. So really, any molecule that's got a CHOH, I mean a carbonyl group, right, you can turn that into you can, at least on paper, turn that into an alcohol using NADH. In this case, this is the alcohol dehydrogenase to form ethanol. So that step right there was this step, right? So the question is, well, where did the ethanol get, the yeast get the acetaldehyde to begin with, right? Well, the reason I got this slide together is to show you two dehydrogenases, right? The yeast will use pyruvate decarboxylase, which we go into in the TPP carbanion shift base slide set, okay, to form acetaldehyde. And that is covered again here. We're not going to go into it in depth here, right? But why don't we just take a quick reminder, tour through this, okay? So TPP is in pyruvate decarboxylase. We know that this is the full structure of TPP. We know that we can shorthand it, R and R prime. We know that it has this positively charged nitrogen and this carbon, making this very acidic, because when you pull this off, it forms that yllid. which this electron sink stabilizes that with a little bit of help from the sulfur. It's mainly the nitrogen now. And that to go from pyruvate to acetaldehyde, if we just look at the reaction of itself, pyruvate to acetaldehyde, we are decarboxylating it and protonating that to an aldehyde. So we break that bond. And anytime you think TPP, think to yourself, make it, break it, make it, break it. That's why, at least what I suggest. And that's what we see here, right? Deprotonation of the TPP to form that yielded carbanion. It attacks this carbonyl group. Those will pull a proton off of an acid to form the hydroxyl group. We then have a rearrangement of electrons where these electrons go here. They're getting pulled to the electron sink. What we're doing now is we are breaking this bond. So we made a carbon-carbon bond, we're breaking a carbon-carbon bond. Also made a double bond here, but really the reason I don't focus on that when I say make it, break it, make it, break it is because although when we go down there it is a double bond, it's in resonance, right? And since the next step is it's in a carbanion form, really those electrons are sitting right there. So make it, break it, and then we're going to go ahead and in this case, we don't make another carbon-carbon bond, but we do make a carbon-hydrogen bond, make it, and then break it. And so then when we look at pyruvate decarboxylase and alcohol dehydrogenase combined, we're talking about alcoholic fermentation by yeast. TPP is used in pyruvate decarboxylase to decarboxylate pyruvate to acetaldehyde. Alcohol dehydrogenase uses NADH to convert that aldehyde into an alcohol. What is the main function of this reaction? It is to regenerate NAD+ so that it can go to glycolysis in times of low oxygen to run glycolysis from glucose to pyruvate to make substrate level phosphorylation ATP, 2 ATPs per glucose. And then what happens during that is that then the pyruvate goes through these and then ethanol can build up. Ethanol can be used by other organisms, and of course humans have certainly exploited yeast to make lots of ethanol. And that is fermentation. Goals of fermentation are what is the purpose, right? Regenerate NAD+. Why? Low oxygen, not running oxidative phosphorylation. Also, some of those molecules are used by a variety of organisms, right? Whether it's propionate, whether it's ethanol, whether it's butyrate, whether it's lactate. In the case of us, another reason for that fermentation step is to take pyruvate that we get from some other source. If we're low on glucose and liver needs to make it, we're not converting glucose to pyruvate to then convert to lactate, to then convert back to glucose, right? If we're talking about gluconeogenesis from lactate, then what we're talking about is getting that lactate from other molecules. So pyruvate we can get from other sources. So we'll talk about that at another time.